Hello everyone, I'm Eber. Today we will be examining if 8 is really the optimal pH for trypsin. So you might ask yourself, what is an enzyme? An enzyme is a catalyst that lowers the activation energy needed for reactions to occur. It does so by forming the bonds of substrate molecules and creating microenvironments that promote the reactions. Most enzymes are globular proteins, and you begin the catalyzation of reactions when an enzyme binds to a specific substrate molecule at an active site. This binding creates conformational change in the substrate molecule that in turn lowers activation energy. This presence of enzymes is so critical to human life because without them, the chemical reactions in our body would not proceed in a fast enough time to meet the needs of human life. This is largely due to the high activation energy chemical reactions in our body require. I hypothesize that as the pH deviates from the optimal pH at 8, the enzyme activity of trypsin will decrease in the breakdown of azocasein, largely because I know that trypsin is the most active in the small intestine when the pH is around 8, which is optimal. And I know that if the pH isn't optimal, it can affect the enzyme by changing its conformation because the environment wouldn't be optimal for its activity. So I asked myself, what would happen if the enzyme, in this case trypsin, wasn't in the ideal environment and was below 8 or above 8? And this is what led me to my hypothesis. So the purpose of my protocol was to test buffers at different pH levels and to examine how enzyme activity changes as it deviates from the optimal pH of 8. I first began by gathering 5 1.5 ml clean centrifuge tubes. I filled the 5 centrifuge tubes with 450 microliters of 0.15 molar sodium chloride and 20 tris ECO to achieve a buffer at pH 6. I added 500 microliters of azocasein and 50 microliters of the enzyme trypsin into each of the centrifuge tubes. The 5 tubes were incubated at 50 degrees Celsius for 10 minutes. After incubation, 500 microliters of TCA 10% was inserted into each of the tubes and mixed. The tubes were centrifuged for 2 minutes at 14,000 grams. The liquid portion of the centrifuge tubes was inserted into 4 separate cuvettes. Once the cuvettes were prepared, the spectrophotometer was cleared using deionized water and set at 440 nanometers. Each of the cuvettes were inserted into the spectrophotometer individually, and the absorbance was recorded for each of them at 440 nanometers. After this whole procedure was done, it was done 3 more times for buffers at pH 7, pH 8, and pH 9. The only thing that changed in the procedure that was done again was the buffers. The azocasein was kept the same at 500 microliters, and the enzyme trypsin was kept the same at 50 microliters. It is important to have a control variable without the enzyme trypsin. Trypsin is an enzyme that breaks down the dietary protein. In the case of this lab, the dietary protein will be azocasein. There are cases in which a protein will begin to break down without the assistance of an enzyme. It is critical to have a sample without trypsin because it shows the scientist if azocasein undergoes a breakdown without the presence of trypsin. It will allow the data to be more accurate because it will inform the scientist if the enzyme is the only factor in the breakdown of azocasein or not. The control samples were created and went through the same process as stated in the procedure in the last slide. The only difference is that the 50 microliters of trypsin is replaced with 50 microliters of deionized water for all the same. What we have here are the control sample averages. As you can remember, these control samples do not contain any trypsin. Trypsin is instead replaced with deionized water. We can see that for pH 6, 7, 8, and 9, there is some sort of activity, which means that azocasein does go on natural breakdown, and that trypsin isn't the only factor that breaks down azocasein. In the next slide, you will see how I use these results to get the true value of the breakdown of azocasein. You might ask, what am I going to do with the control samples? Well, I'm going to use the control samples to find the true values of my trypsin samples. So first, I can take any control sample, but just to show an example, I'm going to use one sample at pH 6, which is my original trypsin sample. I'm going to subtract that against the average for the control samples at pH 6, and that is going to give me the true trypsin value for a sample at pH 6. I can do this for any sample at any pH to get the true value. So what we have here are the results for our trypsin containing samples. As we can see, this is a comparison of NGL product per milligram of enzyme per minute and pH 6, 7, 8, and 9. As we can see that as the pH increases, the product produced increases as well. And as we can remember, this also means that the absorbance is higher. And what this means is that there was much more orange material or a broken down azocasin in the solution as read by the spectrophotometer. We can also see, if we look closely enough, that none of the standard deviations overlap, which shows and proves that pH 6, 7, 8, and 9 are statistically different. So in other words, this means that pH 9 is the optimal pH and the pH where enzyme activity is the busiest because it produced more product. So therefore, rejecting my hypothesis that pH 8 was the optimal pH. For the data shown in the last slide, I conducted a t-test. And with this t-test, it conducts an analysis of two data sets. So as we saw in the last slide, I compared the standard deviations and came out to conclude that they are all significantly different. But what the t-test does is that it takes it a step further and analyzes two data sets. And I know that if the p-value is less than 0.05, then the alternate hypothesis is proven, which is the opposite of what is stated in the first column. So I compared the observance of pH 8 and pH 9, and that came out to prove that it was significantly different. I did the same for pH 8 versus pH 7 and pH 8 and pH 6, which all proved to be significantly different. And what this proves is that the optimal pH for the enzyme trypsin in the breakdown of azocasin is 9 and not 8. So my hypothesis is disproven because if you see in the last slide, the pH at 9 was the highest bar, which proves that that is the highest point in which enzyme activity is present. I would say that my experiment was a valid method to test my experiment. It provided me with the necessary data to prove at what pH the optimal pH was present at. In this experiment, my hypothesis was not proven because it proved that the optimal pH was located at pH 9, not pH 8. The visual representation presented on slide 8 showed that the pH at 9 was the highest with enzyme activity. I went a step further and conducted a t-test which proved that all the data was analytically significantly different. So this proved to me that the bar graph was a pretty reliable source. This also proved to me that pH at 9 is the optimal pH of trypsin. It is very important to have a pH that is optimal for the enzymes in your body. For example, amylase, a enzyme in the mouth that breaks down starch, is ineffective at low pHs and is denatured at high pHs. This is why it is important for the environments to be optimal pHs. If I were to change something for the next experiment, I would change the variability of my experiment. I would add pH 10 and pH 9 to the sample pool. A limitation for me was that I couldn't see if pH of trypsin was truly 9 because I didn't test a pH higher than 9. So I had to assume that the pH at 9 was optimal because enzyme activity was highest at that point. Another error I would fix would be to place my samples in ice to reduce the natural breakdown of azocasin before the experimental time has begun. Thank you for taking the time to view my PowerPoint. Before we end, I wanted to give a special thanks to Dr. Margolis for his guidance throughout this experiment. I want to thank the biology department at Towson University for providing the enzyme lab protocol, and I want to thank James Stegman for providing the statistics intro PowerPoint that helped me with my t-test.